late who's joined. Bacteriophages as vector, the lambda and M13 phage. Bacteriophages are viruses that infects bacteria. Phages are very simple in structure, consisting of DNA or occasionally RNA molecule carrying a number of genes, including several for replication of the phage. It is surrounded by a protective coat or capsid, which is made up of protein molecules. Morphologically, there are two types of structures, the head and tail phage and the filamentous phage. Example of head and tail phage is the lambda phage. An example of filamentous phage is the M13 phage. I'm down to the diagram. On the left side, you have an head and tail phage, which contains an head that is capsid coat that is made up of protein bearing the DNA. On the right hand side, you have a filamentous phage that appears to be filamentous in structure and containing the DNA molecule within the capsid. We are going with the lambda phage first. Lambda phage, going to the structure of lambda phage. As I already mentioned, they are head and tail phage. The head is made up of capsid, which is proteinaceous in nature, bearing the DNA molecule. It has a collar, it has a body sheath and a tail with the base plate. The tail fibers which is present is used for pinning the phage particle on the bacterial surface. And the bacteriophage lambda was discovered by Esther Lederberg in the year 1950. I repeat, lambda phage was discovered by Esther Lederberg in the year 1950. So as we all know, this is the general pattern of the infection cycle, which is basically of 3 different steps. The general pattern of infection, which is the same for all types of phage, is a 3-step process. The phage particles attaches to the outside of the bacterium and injects its DNA into the cell. The phage DNA molecule is replicated usually by specific phage enzymes coded by genes in the phage chromosome. Other phage genes direct synthesis of the protein components of the capsid, and new phage particles are assembled and released from the bacterium. And with some phage types, the entire infection cycle is completed very quickly, possibly in less than 20 minutes. This type of rapid infection is called a lytic cycle, as release of the new phage particles is associated with lysis of the bacterial cell. So it will be very easily in an understandable form when we go into the diagram in the next slide. So with some phages, the entire cycle takes place within 20 minutes. So the release of the phage is via lysis of the cell. That is why they are called as lytic phages or lytic cycle. The characteristic feature of a lytic infection cycle is that phage DNA replication is immediately followed by synthesis of capsid proteins, and the phage DNA molecule is never maintained in stable condition in the host. There is lysis or the breakdown of the bacterial cell and continuous release of the phage particle. I repeat the slide again. The phage infection cycle. The general pattern of infection, which is the same for all types of phage, is a 3-step process. The phage particles attaches to the outside of the bacterium and injects its DNA into the cell. The phage DNA molecule is replicated usually by specific phage enzymes coded by genes in the phage chromosome. Other phage genes direct synthesis of the protein components of the capsid, and new phage particles are assembled and released from the bacterium. With some phage types, the entire infection cycle is completed very quickly, possibly in less than 20 minutes. This type of rapid infection is called a lytic cycle, as a release of the new phage particles is associated with the lysis of the bacterial cell. The characteristic feature of a lytic infection cycle is that phage DNA replication is immediately followed by synthesis of capsid protein, and the phage DNA molecule is never maintained in a stable condition in the host cell. So for better understanding of this slide, like last year in microbial genetics, you would have studied these stages in a life cycle when you have studied what is called as transduction. So stages of phage life cycle. The first step of phage's to the bacterial cell goes with adsorption or pinning. So adsorption, then goes penetration of the DNA molecule or injection of the DNA molecule inside the bacterial cell. There is multiplication or replication of the phage DNA. Synthesis of capsid component takes place. There is assembly of the components and release of the components by lysis of the bacterial cell. This is how you would have studied, and that is what is compensatorily minimized to a three-step process in the previous slide. So now look into this diagram on your right extreme, which is a general pattern of infection of a bacterial cell by a bacteriophage. So the first top diagram, the phage attaches to the bacterium and injects its DNA. So that phenomenon, we call it as adsorption. The second stage, if you see, is penetration of the DNA molecule and replication of the DNA molecule. So for the phage DNA molecule is replicated. In the third step, you can find the capsid component synthesis. There is synthesis of the capsid components and new phage particles are assembled and released via cell lysis. So this entirely goes as a three-step process. Elaborately, if you see, it goes as a six-step or 6 stages of phage life cycle. The adsorption, penetration, replication, synthesis of capsid components, assembly, and of course, release. What happens with lysogenic phages? So lytic and lysogenic phages are the two types of phages we come across. In contrast to a lytic cycle, lysogenic infection is characterized by retention of the phage DNA molecule in the host bacterium, possibly for many thousands of cell division. So lysogenic phages remains within a bacterium cell for generations together. That is what this sentence means. In contrast to a lytic cycle, lysogenic infection is characterized by retention of the phage DNA molecule in the host bacterium, possibly for many thousands of cell division. With many lysogenic phages, the phage DNA is integrated into the bacterial chromosome. The phage particle that enters the bacterium gets integrated with the host chromosomal DNA, that is the bacterial chromosomal DNA. The integrated form of the phage DNA is called as the prophage, remains hidden, that is quiescent, and the bacterium, often referred to as a lysogen, which carries a prophage, is usually physiologically indistinguishable from an uninfected cell because it is having an integrated prophage, but it cannot be differentiated as lysis doesn't occur. The integrated form of the phage DNA, called the prophage, is quiescent, and a bacterium referred to as a lysogen that carries a prophage is usually physiologically indistinguishable from an uninfected cell. However, the prophage is eventually released from the host genome and the phage reverts to the lytic mode and lyses the cell. So that is what happens. Even though it is a lysogenic phage, even though it remains within the bacterium cell, is in quiescent form as a prophage, the release of the phage, the lysogenic phage, takes place by the lysis of the cell. The infection cycle of Lambda is a typical lysogenic phage of this type. Okay, so the Lambda phage life cycle is a typical life cycle of the type. I repeat the slide again. In contrast to a lytic cycle, lysogenic infection is characterized by retention of the phage DNA molecule in the host bacterium, possibly for many thousands of cell division. With many lysogenic phages, the phage DNA is inserted or integrated into the bacterial chromosome or genome. The integrated form of the phage DNA, called the prophage, is quiescent, and a bacterium referred to as a lysogen that carries a prophage is usually physiologically indistinguishable from an uninfected cell. However, the prophage is eventually released from the host genome and the phage reverts to the lytic mode and lyses the cell. The infection cycle of Lambda, a typical lysogenic phage of this type. So for better understanding, you can go with this diagram. What happens? So go to the topmost part of the picture. The lambda phage particles attaches to an E. coli host and injects its DNA. So the diagram has bacteriophage attaching to a bacterial cell, and the bacterial cell bears what is called as its own chromosome, which is blue in color. So the bacteriophage DNA is depicted pink in color. One characteristic feature of lambda phage DNA is within the capsid head, it is linear in form. So inside the capsid head, it is linear. Once it enters the bacterial cell, it circularizes. I will explain you that in a greater detail when we go in for the further slide. It has fast site for it to enable circularization. So that I will tell you when we come forward onto the slides. Now just understand, within the bacterial phage head, that is capsid head, the phage lambda DNA is linear in form. When it enters the bacterial cell, it circularizes. So come to the second top picture. You have the lambda DNA, which is circularizing. The bacterial cell harbors or bears the bacterial chromosomal DNA on its left. On its right, you have a lambda DNA molecule. Integration or recombination takes place between the bacterial chromosomal DNA and the lambda DNA, which is the third down picture, which has blue colored chromosomal DNA and an integrated pink color DNA with it. So the pink color lambda DNA is called as a prophage molecule, and the bacteria that harbors the prophage is called as the lysogen. The third stage, if you see, it is lysogenic form. Now what happens, as we all know, bacteria multiplies by binary fission, one cell divided into two. The chromosomal DNA that bears the prophage, if you see, are equally distributed to the daughter cell. So there is two fates that we need to know. There is two phenomena that we have to consider. The process A and B, which is depicted on the picture, induction. Induction is the phenomenon where you excise the lambda phage molecule from the chromosomal DNA that occurs naturally. There is assembly that takes place and release of the phage particles via the lysis of the bacterial cell. So this phenomenon of induction, that is excision of lambda phage, occurs naturally. We can also induce it artificially by increasing the temperature of growth of the host organism from 37 degrees Celsius to 42 degrees Celsius. So we are increasing the temperature of incubation to 42 degrees Celsius to bring about artificial induction during gene cloning experiments. So I think the picture is very clear. A limited number of lysogenic phages follow a rather different infection cycle. So here we are coming at what is called as the life cycle of M13 phage. A limited number of lysogenic phages follow a rather different infection cycle. When M13 or a related phage infects Escherichia coli, new phage particles are continuously assembled and released from the cell. The entry and exit of these type of phages is via sex pili. So there is no breakup or break of the cell to release its component, that is phage particle. But the entry of the M13 phage is via sex pili. The exit of the phage particle is via sex pili. Said there is no breakdown of the host cell. So the entry and exit of phage is via sex pili, what we call as conjugation pili. The M13 DNA is not integrated into the bacterial genome and does not become quiescent. So as I told you, in case of lambda phage, there is integration. M13 phage does not integrate its DNA inside a bacterial genome. So it doesn't remain quiescent. With these phages, cell lysis never occurs and the infected bacterium can continue to grow and divide, albeit at a slower rate than uninfected cells. So since there is one type of phage entering it, its immune will become a bit lesser, so it will multiply in a slower rate, but it does multiply. That is what this sentence means. With these phages, cell lysis never occurs and the infected bacterium can continue to grow and divide, albeit at a slower rate than uninfected cells. I repeat the slide. A limited number of lysogenic phages follow a rather different infection cycle. When M13 or a related phage infects Escherichia coli, new phage particles are continuously assembled and released from the cell. The entry and exit of phage is via sex pili. The M13 DNA is not integrated into the bacterial genome and does not become quiescent. With these phages, cell lysis never occurs and the infected bacterium can continue to grow and divide, but at a slower rate than uninfected cells. Now look into this diagram, the infection cycle of bacteriophage M13. So the first top picture, you will have an M13 molecule. M13 DNA is entering the bacterial host cell, Escherichia coli cell, via the sex pilus. So now you need to know the M13 DNA is a single-stranded circular DNA molecule. Okay, so M13 specific M13 of interest, if you see why M13 has pulled up interest as bacteriophage vector is because of its circularized single-stranded DNA genetic element. So M13 DNA undergoes replication. There is assembly of the particle. If you go into the third stage of the picture, there is rapid cell division taking place, and phage M13 is equally distributed to the daughter cells, and release of the M13 particle again takes place via sex pili. There is no breakdown of the host cell, that is host Escherichia coli cell. This is one type of lysogenic phage, unlike Lambda phage. Now we are going in greater detail of Lambda DNA molecule as a vector molecule. Gene organization in the Lambda DNA molecule. So this is the Lambda genetic map which you need to know, showing the position of important genes and functions of the gene cluster. So before going to the picture, we will come down for description, then go to the above picture. The Lambda DNA molecule is 49 kilobase in size and has been extensively or intensively studied by the techniques of gene mapping and DNA sequencing. So if you look in the picture now, you start with the 0, end with the 49, right? So the Lambda DNA molecule is 49 kilobase in size. As a result, the positions and identities of all the genes in the Lambda DNA molecule are known. So it is mentioned as E, there should be a term Lambda. So there is a typo error, typing error that has occurred. So in the Lambda DNA molecule are known. So if you look into the picture again, you can see various positions starting from 0 to 20, that is still 20 kilobase. There is regions for capsid components and assembly. If you look into the region 28 to 34, there is integration and excision region. 34 to 40, if you see, 41 if you see, there is early regulation. 42 to 44 if you see, there is DNA synthesis. 46, the region gives you the late regulation, and 48 if you see, the region gives you the lysis of the host that is involved in the life cycle of the Lambda phage molecule. So that is what this sentence means. The Lambda DNA molecule is 49 kilobase in size and has been intensively studied by the techniques of gene mapping and DNA sequencing. As a result, the position and identities of all the genes in the Lambda DNA molecules are known. A feature of the Lambda genetic map is that genes related in terms of functions are clustered together in the genome. So if you look into the picture again, one left extreme, you have capsid component and assembly. On the right extreme, you have various regions that is involved in the life cycle. So the genes are in terms of clustering. So that is what this sentence means. A feature of the Lambda genetic map is that genes related in terms of function are clustered together in the genome. For example, all of the genes coding for components of the capsid are grouped together in the left-hand third of the molecule, and genes controlling integration of the prophage into the host genome are clustered in the middle of the molecule. So if you look into the picture again, you have a region called as B2 region between. They are non-essential region. If you remove that and replace with a foreign gene, you can very well use this DNA as an vector system. That is what this means. For example, all of the genes coding for the components of the capsid are grouped together in the left-hand third of the molecule, and genes controlling integration of the prophage into the host genome are clustered in the middle of the molecule. Clustering of related gene is profoundly important for controlling expression of the lambda genome, as it allows genes to be switched on and off as a group rather than individually. Clustering is also important in the construction of lambda-based cloning vector. Maybe we will go with the slide once again regarding the gene organization in the lambda DNA molecule. The lambda DNA molecule is 49 kilobase in size and has been intensively studied by the techniques of gene mapping and DNA sequencing. As a result, the positions and identities of all the genes in the lambda DNA molecule are known. A feature of the lambda genetic map is that genes related in terms of function are clustered together in the genome. For example, all of the genes coding for components of the capsid are grouped together in the left-hand third of the molecule, and the genes controlling integration of the prophage into the host genome are clustered in the middle of the molecule. Clustering of related genes is profoundly important for controlling expression of the lambda genome, as it allows genes to be switched on and off as a group rather than individually. Clustering is important in the construction of lambda-based cloning vector. The linear and circular forms of lambda DNA. So as I told you when we saw the life cycle of the lambda phage, within the capsid head, lambda DNA is linear in form. When it enters the bacterial cell, it circularizes. So there is two forms of lambda DNA, the linear and the circular forms. So we will see to that in a greater detail, the linear and circular forms of lambda DNA. So look into the picture above. You have two different pictures, A and B, in a single picture. Okay. So the first one, A, depicts the linear form of the lambda DNA molecule. So this is a linear molecule. You have two different regions on the extreme end. That is the right cohesive end and the left cohesive end, what you call as right cos site and the left cos site, and the specific nucleotide that is mentioned is constant. You cannot change and write. On the left cos site you have GGG, CGG, CGA, CCT. On the right cos site you have CCC, GCC, GCT, GGA, which are complementary to each other. That is what we call single-stranded flanking on the either end of the lambda DNA molecule. So within the capsid head, this linear form is found. Once it enters the bacterial cell, the right cos site combines or ligates itself with the left cos site, forming an double-stranded circular form of the lambda DNA molecule. If you look into the picture B, you can see how it is complemented with each other to produce an circular molecule. So the circular form of lambda DNA molecule. So picture A tells you the linear form of the DNA molecule lambda, and B tells you the circular form of the lambda DNA molecule. A second feature of lambda DNA that turns out to be of importance in the construction of cloning vector is the conformation of the DNA molecule. At either end of the molecule is a short 12 nucleotide stretch in which the DNA is single-stranded. Two single strands are complementary. Okay, so those two single strands are complementary to each other and so can base pair with one another to form a circular, completely double-stranded molecule. Complementary single strands are often referred to as sticky ends or cohesive ends because base pairing between them can stick together the two ends of a DNA molecule. So the first important feature of lambda DNA as vector, it is 49 kilobase in size, and the genes regarding functions are clustered together on either ends. That is one advantage. The second feature why it stands to be a very important vector system is because in the head, capsid head, it remains linear, and once it enters the bacterial cell, it gets circularized, acting like a plasmid molecule. The lambda cohesive ends are called the cos sites. So right cos site and left cos site is how it is designated, and they play two distinct roles during the lambda infection cycle. First, they allow the linear DNA molecule that is injected into the cell to be circularized, which is a necessary prerequisite for insertion into the bacterial genome. As I told you, there should be prophage formation, there should be integration. To be integrated with the bacterial chromosomal DNA, first the lambda DNA molecule has to circularize itself. So the first step, they allow the linear DNA molecule that is injected into the cell to be circularized, which is a necessary prerequisite for insertion into the bacterial genome. The second role of the cos site is rather different and comes into play after the prophage has excised from the host genome, what we call as induction. So within the capsid head, it should be linear in form. So one circularized form, to go into lysis of the cell, it has to convert itself to linear form, pack itself inside the capsid head, and then release. That is the second stage. The second role of the cos site is rather different and comes into play after the prophage is excised from the host genome. At this stage, please listen carefully, at this stage, a large number of new lambda DNA molecules are produced by the rolling circle mechanism of replication, in which a continuous DNA strand is rolled off the template molecule. The resulting molecule is a catenane consisting of a series of linear lambda genomes joined together at the cos sites. If you look into the picture, you will extremely understand things. Just know now, at this stage, a large number of new lambda DNA molecules are produced by the rolling circle mechanism of replication. So replication has to take place for multiplication. So it takes place, the mechanism that takes place is the rolling circle mechanism of replication, in which a continuous DNA strand is rolled off from the template's molecule. The result is a catenane consisting of a series of linear lambda genomes joined together at the cos sites. The role of cos site is now to act as a recognition sequence for an endonuclease that cleaves the catenane at the cos site, producing individual lambda genomes. This endonuclease is a product of gene A on the lambda DNA molecule, creates the single-stranded sticky ends, and also acts in conjunction with other protein to package each lambda genome into a phage head structure. So E genome alla, it is lambda genome. Again, insertion spelling typo error. The role of the cos site is now to act as recognition sequence for an endonuclease that cleaves the catenane at the cos site, producing individual lambda genomes. This endonuclease, which is the product of gene A on the lambda DNA molecule, creates the single-stranded sticky ends and also acts in conjunction with other proteins to package each lambda genome into a phage head structure. The cleavage and packaging processes recognizes just the cos sites and the DNA sequence to either side of them, and so changing the structure of the internal region of the lambda genome. For example, by inserting new genes has no effect on these events, so as long as the overall length of the lambda genome is not altered too greatly. So the capacity of capsid head is 49 to 52 kb in size. So unwanted region, the lambda capsid head during assembly identifies or packaging identifies the cos site and the clusters of gene on the either side, the middle portion, if you alter the gene with the foreign gene, it is accountable. That is what this sentence means. The cleavage and packaging processes recognizes just the cos sites and the DNA sequence to either side of them. So changing the structure of the internal regions of the lambda genome, for example, by inserting new genes, has no effect on these events so long as the overall length of the lambda genome is not altered too greatly. So for better understanding, we will go to the diagram, which is what the diagram is all about. So now go into the picture C, replication and packaging of the lambda genome. So the first one, if you see the first topmost picture, you have the rolling circle mechanism of replication. So the entire set of DNA strand with more than one cos site rolling off from the template lambda DNA is called as a catenane. Catenane is a region that has more than one cos site. Okay. Now what happens in picture 2 down? The gene A produces endonuclease that recognizes the cos site. The cos site is for recognition site for gene A product. It cleaves the cos site. After cleaving, you have the linear molecule lambda DNA. On either side, you have left cos site and right cos site. Now what happens is you need to go in for assembly, protein component synthesis and assembly. So each cos site molecules, that is either left, right cos site, and the molecule is inserted into the new phage capsid head and release takes place via the cell lysis of the host cell. Now what do I mean? The package, what you call as in vitro packaging, recognizes what is called as the cos site. It recognizes the cos site and the cluster of gene on either side of the lambda DNA. Central portion is no way accountable. It does not recognize. So removing the gene, inserting a foreign gene inside and packaging to the lambda phage capsid head can eventually be done wonderfully with gene cloning technique until the size of the molecule does not go higher than 49 to 52 kilobase. That is what this sentence and the previous slide meant. So we will go back to the previous slide so that you will again understand. Just look into the slide once again. The lambda cohesive ends are called the cos sites and they play two distinct roles during lambda infection cycle. First, they allow the linear DNA molecule that is integrated into the cell to be circularized, which is necessary prerequisite for insertion into the bacterial genome. The second role of the cos site is rather different and comes into play after the prophage has excised, has induced from the host genome by the phenomenon called as induction. At this stage, a large number of new lambda DNA molecules are produced by the rolling circle mechanism of replication in which a continuous DNA strand is rolled off the template molecule. The result is a catenane consisting of a series of linear lambda genome joined together at the cos sites. Now, the role of the cos site is to act as recognition sequence for an endonuclease that cleaves the catenane at the cos site, producing individual lambda genomes. So the endonuclease is the product of gene A on the lambda DNA molecule, which creates the single-stranded sticky end and also acts in conjunction with other proteins to package each lambda genome into a phage head structure. The cleavage and packaging processes recognizes just the cos sites and the DNA sequences to either side of them. What do I call as cluster of gene? So changing the structure of the internal region of lambda genome, for example, by inserting new genes has no effect on these events so long as the overall length of the lambda genome is not altered too greatly.